Hello, I'm Travis Baer, the Chief Technology Officer of Sound Agriculture, and I'm here today with Marie Pearson, a Product Manager at Sound Agriculture, and we're going to tell you about a new product that we are launching called Source. We want to take a step back and tell you about how we see the problems in agriculture today. One of the issues we see is that growers are leaving extra bushels in their field every year because they don't have the tools to unlock the soil's full potential. Growers work very hard and have a lot of good tools to deal with things like disease and insects and weeds, but don't really have a lot of tools to unlock the potential in the soil. We know that soil is really important for plant health and for yield. It has a rich array of microbial life, and that microbial life is involved in making nutrients available to the plant. So this is the backdrop to our development of the Source product. What we wanted to do here was develop a product that could unlock that potential. And this product really relies on some breakthrough scientific discoveries that we've made in the lab about how plants interact with the soil microbes. We can build on these discoveries and translate that to a product that's going to provide an economical and sustainable solution for unlocking nutrients in the soil and providing more to the plant and more yield to the grower. So how does Source work? Well, if we think about the soil and root microbiome life, what we have is a situation where the plant is communicating with microbes, and this communication is happening so that the plant and microbe can exchange nutrients and cycle nutrients through the soil. What we figured out how to do is to stimulate that conversation, to basically add chemistry that we design in the lab to that system and enhance the natural metabolic activity of those microbes. We're focused on two different types of microbes here. One is nitrogen fixers. These are microbes that are able to turn atmospheric nitrogen into plant-available forms, as well as phosphate solubilizers, which are able to unlock phosphate that might be tied up in the soil. So effectively what we're going to get with this product is a system that manufactures nitrogen and phosphorus fertilizer right at the root zone. So I'm going to show you a little bit of data from the lab and from the greenhouse that gives us confidence that we have a good understanding about the mode of action here. So again, we are adding chemistry to the system that stimulates the natural metabolic activity of these microbes. So what you're looking at in this slide is an assay where we can measure the increase in nitrogen fixation genes in the microbes we are looking at. And so with Source, we see a significant increase in gene expression in those microbes. This is a similar study, but here we're looking directly at the nitrogen fixation activity. This is measuring the enzymatic activity of those microbes to transform atmospheric nitrogen into a plant-available form. And what we see that as we add more Source, we get more of this beneficial activity, which means more nitrogen for the plant. We also see some interesting effects when we look at an entire soil consortium, when we look at the millions of species that are available right at the root zone. In control-treated plants, we see this amazing diversity of microbes there. Some of these are beneficial. Some of these are not doing much in terms of metabolic activity for the plant. What we see when we add Source, though, is that the abundance of the nitrogen-fixing bacteria in that community right at the root zone is increased. So not only are we increasing the activity of these bacteria, they're also more abundant right at the root zone. This means that, again, more nitrogen can be transformed from the atmosphere into a plant-available form to drive plant health and yield. Just to talk about the phosphate side of things, we see a similar effect when we look in the lab and in the greenhouse at phosphate solubilization. So what you're looking at here is an experiment where we can measure directly phosphate solubilization in a model microbe. And what we see is significant increases when we add the Source active ingredient. So again, this is providing a boost of phosphorus right at the root zone that the plant would not otherwise be able to access. Looking at microbes and looking at the soil consortia is really interesting for us as scientists, but as we translate this to the field, we really want to know what happens in terms of plant health. So what we're looking at here is some experiments from our lab and greenhouse where we can measure aspects of plant health and plant productivity, including biomass, greenness, and indicators of plant stress. What we see across the board is that when we add Source, we see greener, healthier, and larger plants. And this really gives us confidence that this is going to translate into a yield increase in the field. So to summarize on the mode of action, again, this is chemistry. So we are stimulating the native soil microbiome. We're turning on nitrogen fixers, so that's going to increase the available nitrogen to the plant right at the root zone. we're increasing the activity of phosphate solubilizers so we can unlock phosphate that's otherwise tied up in the soil. And both of those together add up to plant health and to increase yield.